All right, everyone. Once again, welcome back. Good afternoon, and welcome to everyone who's viewing from home from the recording. This will be the last in our guest lecture series for 2025. We will start up again in early January, and I hope to have all of you in audience when we do so. But perhaps we've saved the best for last. I'm super excited for today's talk. Our guest speaker is volunteer Graham Kelly, who will be talking about transgenics. I imagine a lot of folks in the audience today have heard of things like GMOs, genetically modified organisms, especially in the context of agriculture and food. You've likely heard of cloning as well, but transgenics maybe not as well known. It's in the news increasingly though. So today's a great chance to learn more about it. So with that, Graham, I'll turn the Zoom over to you, and I look forward to learning more about transgenics. Thanks, everyone.
Okay. Hi there, everyone. My name is Graham Kelly, and I'll be talking about transgenics today. This is my third lecture here at the museum, and I'm really excited to show you guys a little bit about transgenics. I assisted in some transgenic work at Rockefeller University on some de-extinction projects, and I'll get into that a little bit later. But let's just start off with transgenics. What is it? Sorry. Today we're going to be focusing on its applications for conservation projects around the world. But get it out of the way, what is transgenics? Well, what is it? Transgenics is the transfer of a gene from one organism to a host organism to give that host organism whatever trait that gene confers. So scientists taking one gene, inserting it into a host, and giving that host a trait it wouldn't normally possess. It's typically done between species, but also can be done between members of the same species or different breeds of the same species. And it's really based on some ancient tools of breeding that have been used for centuries that have been made more efficient with modern scientific tools. So, sorry, I'm going to give a little example. This is a transgenic organism I made last weekend. And what's interesting about this, I wanted it to be an example about transgenics. These are two slides filled with bacteria, or sorry, two petri dishes filled with bacteria. And if you look closely, you're going to see some green, some red, and some colorless dots there. And those are colonies of bacteria. But like I was saying, transgenics is transferring a gene from one organism into another to give it whatever trait that gene controls for. So for us, if we look on the left, we can see those red dots that are spaced out, and they're really bright. It's very substantial. What those are is bacteria, like you're seeing with all these other colonies next to them, but those specific ones that are red are transgenic. There is a gene that exists within coral reefs, and this gene allows the coral to have a red bioluminescent coloring. And when you take out that gene for red bioluminescence and insert it into a bacteria, you can give that bacteria the coral's ability to bioluminescent red. If you look on the right, the same thing is happening there, but instead of a transgenic organism that's bacteria and coral, the one on the right is bacteria and jellyfish, because some jellyfish can bioluminescent green. And if you put that gene for bioluminescence into these bacteria, they're going to glow green. So what you're seeing is a bunch of colonies of bacteria with some transgenic individuals springing up here and there. And what's noticeable about them is that they're bioluminescent. So this is an example of a transgenic organism. So what we're going to do today, these are going to be our core topics. We're going to talk about how transgenics is done, how we can use it for conservation. We're going to discuss the question of should we use it and the risks that come along with using it. And then we're going to wrap up with a group discussion. And I'd really love as many people's involvement and participation. There's no wrong answers. This is a big topic of ethics, and there's a lot of pros and cons for both sides. So I would love to hear everyone's opinion at the end. So let's start off. How did the idea of transgenics originate? Well, like I said, it was based on more older breeding principles. And I'm going to give an example of how this would work. Let's say I have a bunch of dairy cows, but I want to make a cow that is more adapted to the heat, something that I'm going to move to a more arid environment, and I want a dairy cow that can still produce a lot of milk. So one thing I could do is find a stock population in an arid environment and take those cows that live in that arid environment and only breed the ones that really do well in that increased heat. And I would not breed the ones that struggle in the heat. Over generations, you're going to have a breed of cattle that is very heat acclimated, very adjusted for that warm environment. And then I could take my dairy cows and breed them with these heat adjusted cows, and their offspring are going to possess those genes for heat resistance, hopefully. So this is a, pretty standard and ancient breeding practice that has been done for centuries. But what happens nowadays is that we can accelerate this process with different genomic tools. And I'm going to get into that in the next slide. How is a transgenic organism made? So the first thing you would do is you would try to identify a gene responsible for your selected trait. So for our cow example, I may go to that stock population of heat acclimated cows and try to find the gene that allows them to be so heat acclimated, allow them to resist the heat. Once I isolate that gene, I'd want to amplify it, which means make a lot of copies of it. I would then insert that gene into the host embryo. So insert that heat adjusted gene into the embryo of a dairy cow. And I can do this using CRISPR or a virus or injections, very micro injections. After that, I would implant the embryo into a surrogate because you have to remember this is a transgenic organism. It's not simply a dairy cow. It's not simply one thing. It would have to be implanted into a surrogate. And we're going to get a little more into that later. But after it's implanted into a surrogate, I would raise that individual once it's born and breed it to see if it can pass down those transgenic traits. So my dairy cow is being edited with heat acclimated genes, and it's then going to have offspring that hopefully are heat acclimated and possess that gene. But I can test this by reproducing that individual and seeing if it's truly transgenic, because we want to make sure that what we're doing is a permanent alteration. We want to make it so that the offspring also have this transgenic gene, which means the gene has to get into its germline or sperm and egg so it can be passed down to offspring. And this is some of the work I helped with, which was in a kind of lab collaborating with Colossal Biosciences. I'll also get into them later. But yeah, we were testing to see if these traits, these transgenic genes, were being passed down. So now that we know roughly what transgenics is, how can we use it for conservation? Well, we're going to get into three main topics today. We're going to talk about gene drive on islands with rats. We're going to talk about assisted evolution for corals and toads. And then we're going to wrap up with de-extinction with the thylacine. Gene drive. What is it? Gene drive is a genetic tool that helps a gene spread through a population by increasing the likelihood that that gene is passed down to one's offspring. So instead of a 50% chance, we can use different genetic tools to ensure, we can use CRISPR to ensure that the offspring are 100% going to get that trait. Instead of 50-50, we're going to be certain about it. And by doing so, we can easily and quickly spread a gene through a population. So if we have a helpful gene that gives a helpful trait, then we want to really quickly spread it through a population. So we would use gene drive. And then why would we use gene drive and where would we use it? Well, we'd want to typically use it on islands. And I'm going to give you a little information about why islands are so important and why gene drive is so useful for islands. So to start off, islands possess only 5% of Earth's land area, but control for 20% of its biodiversity. We have 75% of global extinctions happening on islands. And if you go on any given island, up to 90% of the vertebrate organisms on that island can be endemic, meaning they only exist there. So I'm going to repeat that. That's up to 90% of the organisms there live only there on that island. So it creates a real biodiversity hotspot. These islands possess so much biodiversity that it's no shock that 50% of the threatened species across the world live on islands. So that's 50% of threatened species on 5% of our Earth's landmass. So I'm going to give a little example about why gene drive is so effective to help island environments. Like I mentioned, islands are biodiversity hotspots. How does this happen? Well, when an island springs up out of the ocean, a volcano makes a little island, some plants may drift over there, but the first animals that the real pioneer species that come to these islands come through winds or through storms, such as birds, bugs, and flying mammals. A bird or a bat may be migrating or moving to a different environment, and a big gust of wind sends it out to sea, and it can find its way onto these islands where then it's going to establish itself. We also have bugs and maybe small animals and plants that can drift in during a tsunami on a piece of driftwood and also establish themselves on islands. So any island typically has its first species being a lot of birds, insects, and flying mammals, if any mammals. When all these species come to this island, it's a untapped resource. There's so many different niches to exploit. There's so many different food sources to use and so many environments to manipulate that we're going to have this increase in biodiversity as these few birds, bats, and bugs really diversify to fill every niche. And that creates so many new species, which is why these island environments have such high biodiversity because the animals there are allowed to diversify without a threat from other animals coming in and wiping them out. But what threatens this biodiversity hotspot? Why are islands so under threat? Well, a lot of the times we've seen throughout history, maybe European travelers or traders will come to islands with a lot of grain and supplies. And when they come here, their boats contain a lot of rats. A big boat journey is going to have naturally a lot of grain in the hull of the boat, and that grain brings rats. So when that boat lands on this small island that's really been untouched and isolated for so long, these rats come out on this island and say, hey, there's this untapped resource, all these animals that have no adaptive skills to overcome me. There's no predators that know how to eat me, and there's no prey that know how to avoid me. So something as simple and small as a rat finding its way into an island can really decimate that biodiversity hotspot. These rats can eat the eggs of the birds, wiping them out. They can eat the bugs, wiping them out, and they can eat the bug species that the bats eat. And over time, these rats on their own can really crumble that ecosystem by taking out its main players and all the essential animals that live there. And by doing so, it causes a collapse of that ecosystem. The equilibrium is gone. And over time, it can't even support the rats that came there, the rats that caused all this destruction. And over time, that whole ecosystem is going to collapse. So this is what's happening on these islands. This is why they're so under threat globally. But how can we use gene drive to stop this? Well, let's go back to the start of this problem. Rats have just made their way to this island, and they're about to exploit all the resources of this island, and they're going to do so unchecked. But what if we introduce a transgenic rat? And this transgenic rat has a coded CRISPR gene in its DNA. This is a transgenic gene we've inserted into its genome that makes it so that when this rat, and this is one we're bringing to the island, when this rat breeds with the invasive rats already on the island, it passes down 100% of the time an infertility gene. And you may wonder, how does that spread through the population? Well, it breeds males who are infertile, and it breeds females that are fertile but can only give birth to either infertile males or other females that, once again, only breed infertility. So when this gene is introduced to this population, you can just get one individual to come into this huge population of invasive island rats, and it's going to 100% of the time spread its gene. So that gene will move throughout the population and spread to eventually all the rats on the island, and is going to, over time, wean that population down because of this infertility until there's no rats left. And over time, they will be wiped out, and the native organisms that live on this island, that have evolved on this island, can once again reclaim dominance and diversify, bringing about the biodiversity hotspot that was once there. So this is one method. We're now going to get into assisted evolution. And I hope everyone knows what evolution is and how adaptation works. If anyone has any questions, please let me know. But with assisted evolution, what we're doing is humans are helping these organisms adapt faster than they could normally on their own by speeding up natural processes. So this can look very different. It can come about in a lot of different ways. We can selectively breed individuals with helpful traits. We can expose individuals to stresses they may face in the environment, but do so in a controlled environment so that they can more easily adapt and evolve to the challenges that they're facing. Once we've done that, we can add genes from those evolved or bred individuals into the wild population, giving them these genes that would take millions of years to evolve, but giving them to these wild organisms in a much quicker way. And we can use assisted evolution to help ensure species survival in the face of so much ongoing environmental change. that's happening in the natural world. So basically, it's a way to help organisms adapt to the quick change that's happening in the world. Usually, adaptation takes, thousands, millions of years. But if we accelerate it, we can help these organisms make those millions of years of evolution in a short time and survive in the modern world. So let me give an example of how this would work. We're going to talk about coral and toads. First, coral. So let's take a look at this coral reef right here. A coral reef is another biodiversity hotspot. It supports a nursery of life where there's a lot of young fish or turtles or a lot of different organisms that rely on coral reefs to raise their young. Coral reefs are really important environments and the oceans depend on them. But with increased global change, we're having more sun exposure, more high temperatures in the ocean, and increased salinity in the ocean. And all these increases in temperature and salinity affect the coral. If you don't know, corals are not one organism. It's a colony of organisms that have a very delicate relationship with one another. And if they get uncomfortable by increased temperature or salinity, they're going to bleach, which means basically die out. And corals are facing increasing events where there's high salinity and high temperatures in their water. And it's bleaching the coral and turning them white, killing them. So this is a natural process that would happen. Bleaching events do happen naturally, but they're supposed to take course every 15 years or so. And it takes, or sorry, every 10 years or so. And, it takes about 10 years, 5 to 10 years to recover. Usually the environment would be able to recover, but if these bleaching events are happening more frequently, then it's not giving the environment a chance to recover and rebound. So we need to help these coral evolve to an increased change in salinity and temperature. How can we do that? Well, this coral bleaching is going to wipe out that biodiversity hotspot. So what's our solution? So we can take these corals and bring them to a controlled environment, which you see here. There's a lot of different tubs of water, and we can put a bunch of different coral all in these different tubs. And we are going to expose those corals to a 1 degree increase in temperature. And we're going to see which of all these individuals survives the best, which one keeps growing, isn't wiped out by that temperature increase, but is able to still persist through it. And once we find which individual can best survive, we're going to selectively breed that one. We're going to breed him and create a lot of offspring. And then we're going to expose those offspring to an increase in temperature and salinity. We see which one thrives the most and then breed that one. And we keep doing this and repeating this process until we've created the coral on the left that possesses genes for resistance. And that coral, it has this gene for resistance, and we can take that gene and move it into any coral species we want, giving any member of a coral reef resistance to increased salinity and temperature change. If the whole coral reef possesses this transgenic gene, then when a bleaching event occurs, that whole coral reef is going to be able to survive it. It's going to be able to persist, and eventually that biodiversity is going to return. And once again, a biodiversity hotspot will exist. But can assisted evolution be used in other ways? Can it be used to not just adapt to changes in the oceans, changes in our environment, but it can be used to reverse those changes? So we're going to talk about the example of the cane toad. And this little island right here, we're going to call it Australia because cane toads are an invasive species in Australia. What's really damaging about them is that they eat everything. They eat bird eggs, bugs, anything they can get their hands on. So they will really wipe out the ecosystem. But what makes them especially deadly is that they can not only wipe out all the prey species, but they can wipe out the predators. So we'll take the quoll, for example. The quoll is a major predator across Tasmania and Australia and is kind of like a marsupial cat. The quoll would naturally, as a predator, it eats frogs and toads native to Australia. So when it may be approached by a cane toad, an invasive species, it'll eat that cane toad. It'll give it a sniff, take a bite, and eat it. But it's not going to feel good afterwards. What's happening is that cane toad has something called bufotoxin it secretes. These cane toads along their stomach and abdomen have glands that secrete a toxin that is deadly to most animals that eat it. So the cane toad comes into Australia, eats the prey species, and kills all the predators that try to eat it. So it's a species that really goes unchallenged. It wipes out its predators, it wipes out the prey, And what you're seeing in Australia is the cane toad population booming, with the predator and prey species decreasing. Little by little, Australia is becoming an island of toads. What assisted evolution brings into the picture is that instead of letting this process occur where the environment is going to crumble and that biodiversity hotspot will be lost, what you can do is you can edit the predators. You can make them transgenic. How does that work? Well, cane toads are native to South America. Invasive in Australia, yes, but native in South America. And if you go to South America, there's something called a serpent eagle. And these eagles have evolved over millions of years to have genes that convey resistance to bufotoxin. So, through millions of years of evolution, this eagle has adapted to eat the cane toad in its native environment. If you bring that eagle into Australia, you could bring it in as a new predator to hunt the cane toads, but then you're left with the same issue of, okay, the cane toads are gone, but now we have the serpent eagles, another invasive predator. So what can you do? You can go to that serpent eagle's genome and find the specific gene that allows it to digest bufotoxin. You can take that gene and put it into the quoll. By putting it into the quoll, you've made it so that that quoll can happily eat all the cane toads on the island. Not only are you freeing all the prey species from this cane toad, but you're giving a nice snack to the quoll. Instead of having to, wipe out the cane toad, you're creating a new food source for the quoll. And eventually, over time, the quoll is going to wipe out the invasive cane toad on its own. It doesn't need constant years of human intervention and humans culling the cane toad population because that relies on funding, that relies on a lot of, things that humans can't do, which is consistency. But with the quoll, you've made it so that this predator on its own can take care of the problem. You've made it so that now that gene is in the population and the quoll is going to wipe out all these cane toads. So an island of toads is now left barren. And the native animals that once lived there can once again breed, can grow, can live, and that biodiversity hotspot can be reestablished. The next topic I'm going to be talking about is de-extinction. It's a hot topic that a lot of people are talking about, and it's mostly centered around Colossal Biosciences, a company that's trying to bring back species like the thylacine, the mammoth, the dodo bird, a lot of different animals, and it's a controversial topic. So I'm going to show you guys a little animation I made that kind of describes what de-extinction is and how it's being used, and I'm going to show you how this is a transgenic process. Right. In 1936, Russell, could you can confirm if you hear this? Benjamin, male.
We can hear the audio, but we don't see anything on the screen if that's appropriate.
Oh,
I
huh.
think your slides are still up, and it's only the PowerPoint window that's being shared right now, it looks like.
Okay, give me one moment to share this. Can you see it now?
Looks good.
All right. So this is going to explain a little bit about de-extinction. In 1936, Benjamin, a male Tasmanian tiger, was accidentally locked out of his enclosure on a cold night and sadly froze to death. At that time, the Tasmanian government had finally put the species under protection, but it was too late. Then, the last of the Tasmanian tigers was extinct. After the loss of this apex predator of Tasmania, the ecosystem partially collapsed, with prey populations growing unchecked. The tiger was a strong carnivore with a jaw gape of 120 degrees, yet little is actually known about its behavior. It wasn't even an actual tiger, with the sole similarity being their shared stripes. The Tasmanian tiger was a wolf-like pouched marsupial native to Tasmania. The loss of this species was terrible, but maybe not permanent. With the development of genetic engineering tools such as CRISPR, science have begun reassessing how permanent extinction really is. In the early 2000s, efforts began across the continent to revive its former apex predator. It wasn't until the discovery of a 100-year-old improperly preserved joey in the Melbourne Museum that actual progress began. Due to ethanol being its sole preservative, the joey's DNA was only minorly degraded, with strands as long as 1,000 base pairs being found. At this length, the genome could finally be reassembled and was sequenced in 2017. With the genome salvaged, what comes next?
The next issue arose as to how to actually create a specimen with no living individuals today. The closest living relative of the tiger, the numbat, shared their last common ancestor 30 million years ago, yet are still 95% genetically identical. It would take much time, effort, and funding to reconnect these long lost cousins, but it is possible. By replacing sections of numbat DNA with the appropriate tiger DNA from its sequenced genome, a thylacine coated numbat embryo could theoretically be made.
So I'm going to say one thing. Keep in mind this is numbat and thylacine. So the thylacine that they're trying to de-extinct is not a pure thylacine. It's a transgenic organism that has both thylacine DNA and numbat DNA. So when we talk about any extinction project around the world, we have to realize that there's no living individuals today to base our genome off of. So when we do de-extinction projects, we're splicing together the genome we're able to find through fossil records and whatever tissue we can find preserved. We're splicing that DNA and those fragments together with an existing species genome. And that is a transgenic organism because they have genes from both of these species.
So, while this all may be possible, is it the right decision? And what would we even be bringing back? Man made thylacine may not be a perfect recreation due to so much genetic tampering, yet it would only really need to occupy the same niche it formerly did. At the loss of the apex predator, the Tasmanian ecosystem had restructured itself with the Tasmanian devils on the top. As of late, though, a condition known as devil facial tumor disease has made things complicated. This disease is a devil specific contagious tumor that is very easily spread and fatal, currently decimating the devil population. If the tiger was to be brought back, though, not only would it retake its place as apex predator and create stability, but also wipe out devil facial tumor disease by wiping out sick devils before they have time to spread the disease to others. So, is the risk, time, and money worth possibly saving Tasmania, or should we focus our resources elsewhere? Well, I don't know. What do you think?
All right. So that was, sorry, I am still going to be sharing my screen. Let me just go back to my presentation. All right. Oh, okay. So we just showed a little bit about Tasmanian tigers or the thylacine. Both are the same animal. Tasmanian tiger, thylacine, same thing. But you see there how that de-extinction project is essentially just making a hybrid, a mix, a chimera between two species. And that's kind of what transgenic is. It's mixing genes from one organism with the genome of another. And now that it's a possible science that exists in the world, we can do gene drive, we can use assisted evolution, and we can de-extinct long lost species. But just because we can, we have to consider if we should. There's a lot of risks that come with this process, and we have to understand these risks before we take on such a monumental challenge like this. So right now I'm going to go over some of the risks, and I want everyone to consider these risks for our discussion so we can weigh, are the pros worth the cons? Is this something we should consider? Because I'd really love a discussion after this. Um, all right. So I'm going to go back to my previous example of the quoll. So the quoll is a predator in Australia, and let's say we've made it a transgenic organism. We've edited it so it can digest bufotoxin, and it's wiped out all the invasive cane toads. It's become the hero of its island. But just like rats can come to these islands off boats, animals can leave these islands off the boats. So if this transgenic quoll was to somehow get on a boat and wind its way up in South America, where the cane toads are a native species, a natural part of the environment that's required for the environment to have stability. If that quoll was to ever make its way to South America, just like how rats are invasive into islands, the quoll could be an invasive species on its own. And it would be almost a hyper predator because it has such a specialized ability to eat these toads. And if that quoll ever escaped its containment in Australia and somehow made its way to South America, it could decimate that native population of cane toads because it's only really targeted to cane toads. It has no, as an invasive species, it's not really adapted to the native species of that island, but it's surely adapted to eat cane toads. And if it wipes out those cane toads and becomes kind of the top big predator of that environment, then you have the issue of it's wiping out a native species. What was supposed to originally be a transgenic organism that would, remove invasive species, it itself is becoming an invasive species if it ever was to leave that island. It's a really dangerous thing because this is a breed of organism that doesn't really have a place in the world. After the cane toads are gone, it has no environment that it's still suited to be a quoll and exist in Australia, but it's dangerous if it ever escaped Australia. It could wipe out that ecosystem. Oh, sorry. It could wipe out that ecosystem. And just like the cane toads almost destroyed Australia, the quoll in return could wipe out a lot of the native populations in South America. There's also the risk of contaminating our wild population. And we saw with those rats that we can put in a transgenic gene in rats that makes them breed infertility, that makes them spread throughout the population a lot of infertility genes. And rats, they exist all throughout the planet. Rats are everywhere. And on small islands, they can be an invasive species, but on most of the world, rats are a natural native species and, are essential for any environment across the globe. So if this transgenic rat somehow made its way off of these island environments, it came into these islands on a boat and it can just as easily leave these islands on a boat. And if it winds up in a different part of the world that has a native rat population, then, those rats are pretty genetically similar and it could easily spread its infertility gene throughout the population. And just like it did on small islands, it would decimate the rat population, but do so in an environment that needs a rat population. And over time, it would wipe out that native rat species until there's none left. And once again, that ecosystem collapses. Our third risk we may encounter is making populations that are less genetically diverse. And we're going to go back to our coral example. So let's say we've made this transgenic coral and we've made this one individual that has a resistance to changes in salinity and changes in temperature. This coral has an advantage over the not genetically engineered, the not transgenic coral. This coral is able to really rise to prominence in its environment because if a bleaching event occurs, it's going to wipe out all the non-transgenic coral, leaving only the transgenic coral. And with that transgenic coral only being the one, or sorry, with that transgenic individual being the only one left, it's going to reproduce and it's going to fully fill out that ecosystem once again. It's going to reestablish a coral reef because it is resistant to these changes in temperature and salinity. And that's great, right? It seems like that's the whole purpose of what we're doing. But you also have to realize that then that whole coral reef descends from one or only a few individuals. These few individuals have just kind of fully taken advantage of the advantage they have. They've taken advantage of their transgenic gene and they've thrived in changing ocean salinity and temperatures. But that means that the whole reef is descended off only one or two individuals, which means they're all very genetically similar. Which then, if let's say there's a virus that would, the ancestral transgenic coral that we first released was, had a certain susceptibility to certain types of viruses. Everyone's different. We all are more prone to certain things than others. Some people get a flu more often. Some people get, all different infections people are predisposed to. And if those few original transgenic coral we released into the environment had some susceptibility to viruses or anything really, then a virus that came by that would usually only just kill that one ancestral individual can now wipe out the entire reef because they're all descended from that ancestral individual. The whole reef is so genetically similar because they all descend from the same transgenic organism. If these viruses came into a coral reef or a transgenic coral reef, they would usually, before this alteration, just wipe out a few individuals in the reef. But now with transgenics, the reef is so homogeneous that it can really wipe out everyone. Everyone is susceptible to these viruses, and a virus that would usually just be, a agent of change in a coral reef, wiping out some individuals, will decimate the reef and will once again take away that biodiversity hotspot. So we're seeing how transgenics took a coral reef that was crumbling, brought it back to life, only to fizzle out again. Our fourth risk is that we could be returning an extinct species, like the thylacine, to an environment that can no longer support it. What do I mean by this? Well, there's the question of will the prey still recognize their predators? You have these adaptations, these instincts that tell you, oh, that's a thylacine. I need to run away. If you're, sorry, a Tasmanian devil, you know to run away from a thylacine. But if it's been generations since you've seen a thylacine, that instinct can go away if you're not using it. And these Tasmanian devils that would usually be a prey item for the thylacine are now completely defenseless. And what came off as originally the idea being reestablished, put back in a apex predator and reestablish the balance in that ecosystem, you're not bringing back stability, you're making a new invasive species. Because that thylacine, it still knows, it still has that instinct to hunt and eat Tasmanian devils. But these Tasmanian devils don't know to fear a Tasmanian tiger or a thylacine. So you could essentially create a new invasive species. The other question or the other issue you may face is that has the former niche that animal occupied has the former niche been occupied or is it still available? Going back to that example of the thylacine, it has partially had its niche as apex predator taken up by the Tasmanian devils, and we've seen it again, the quoll. The quoll in Tasmania has become kind of an apex predator, and it now hunts a lot of the Tasmanian devils. But if you have an animal that started hunting the Tasmanian devil and you bring in a new predator that's supposed to also hunt the Tasmanian devils, what's going to happen? Well, they're going to compete. You're going to make it so that the Tasmanian tiger or thylacine and the quoll are in direct competition for a food or a resource. And when that happens, instead of adding in an apex predator, you're basically adding in an apex predator and taking one out. So if you make them in direct competition, one of them is going to suffer. Either the quoll population is going to go down, or the Tasmanian tiger or thylacine population is never going to establish itself. They're going to have to compete. And when that happens, you're having two, threatened apex predators competing with one another. Everybody loses. Lastly, we have the question of, do we know exactly what genes we're using? If anyone's ever seen Jurassic Park, that's a good example for this. In Jurassic Park, they de-extinct dinosaurs, but they use transgenic genes. They splice in dinosaur genes into a frog genome. And the error that happens, what really goes wrong, is that they don't realize that the base genome, the frog genome that they're using, has a lot of genes they don't recognize. We're putting these dinosaur DNA on a frog genome, and we don't realize, oh, that's giving the dinosaurs an ability to asexually reproduce. I'm only referencing the movie for those who understand. I can explain it later more if need be, but you don't know what you're putting into that genome. So much of our genome, even with humans, is called junk DNA because we don't know what it does. We don't understand it. And if you're playing with these big genetic tools you don't understand, you don't know what you could be putting into that animal. With Jurassic Park, they were accidentally putting in the ability to asexually reproduce into dinosaurs, making them a huge threat to people. Just like the Tasmanian tiger, what if you accidentally put in genes you don't realize allow it to grow quicker or grow fangs or grow wings? I'm only kidding, but I mean the general idea is still there. You don't know what you're putting in because genetics is something we still barely understand. You're playing God and you don't exactly have a full understanding of what you're doing, and there can be a lot of poor side effects. So these are some threats that come about or risks when you try to tamper in, when you tamper in transgenics. So let's discuss. What are you guys thinking? How do we feel the pros weigh against the cons? I have four topics that we could go over in discussion if anyone wants to choose one or choose their own. This is just something we can go off of if anyone has any thoughts. But some questions that arise from this process is who is responsible for these transgenic organisms? Is it the government? Is it the scientists who make them? Or is it the native people who, lost a key member of their ecosystem? I think the second question, which is one I find most interesting, is how does this impact other conservation projects around the world? And by that, I mean, if you're editing coral reefs to be resistant to temperature and salinity changes, that's great. Coral reefs can persist and still exist in the modern world. But the danger of that is if we can make coral reefs adapt to change, then why stop dumping waste in our oceans? Why stop releasing fossil or using fossil fuels? If we can make any challenge overcome easily by these animals, then why stop throwing challenges at them? You could have a future where, you know, tigers still exist in 1,000 years from now. They haven't been wiped out, but it's only because we've given them the ability to eat food out of a garbage dump. You could be making a species that still persists, but in a world that is increasingly becoming terrible. Why would I ever donate to projects or fight the dumping of pollutants into the ocean? Why would I ever fight that if, the coral reefs that would be threatened are fine? The next question is, when is assisted evolution excessive? If you're making these coral reefs resistant to temperature and acidity change, why not make them immune to anything? Why not make them grow even quicker? That would be great because coral reefs could easily bounce back across the globe. They could reestablish themselves. But what if you've made a coral that too quickly spreads? You could accidentally, once again, create an invasive species. Lastly, there's the question of, is this risk worth it? Is all these efforts worth it if we're just kind of putting a band-aid on the problem? We're forcing those affected by these issues to themselves adapt instead of us having to stop making the problem in the first place. Some could see transgenics as a solution to all the problems we're facing in the modern world, but some could also see it as a distraction from the real issues. So I would love to hear everyone's thoughts, and I really want to thank everybody for their time today. It means a lot that people showed up, and I'm really grateful for it. So I'm going to leave these discussion questions up if anyone has something to say. But yeah, I just want to thank everybody. Thank you very much.
And Graham, thank you also for this talk. I already see one hand in the participants list. If you'd like to jump in and maybe share some of your thoughts about these questions, the ethical concerns of transgenic organisms, please do press the raise hand button. I'll help you unmute and you could jump right in. Looks like Jane will be first. Go ahead, Jane.
First of all, thank you, Graham, so much. I tend to think risks are more worrisome than
Yeah.
although there's some good. My
Mm,
question, though, I'll have to think about it more and listen to others, is about getting, and I don't know if it counts as transgenics,
mm,
but getting a cloning before something is extinct, but is close to it,
mm.
where there's a very few left of something, and it's still in an environment where it sort of fits.
Yeah.
It's still open to our decision, but I'm just wondering how that might fit in and if you have any thoughts.
That is a phenomenal question because you really hit something that's actually happening in the world. The colossal biosciences company that is trying to de-extinct these species, they realize themselves that they're, in a way putting a band-aid on a problem instead of targeting the solution. And I appreciate that this, biotech company is, I'm not a spokesperson for them, but I appreciate what they're doing because they're also preserving the genomes of, like you're saying, species that are close to extinction. They are trying to preserve those genomes so that we don't have to make a transgenic organism. We can know exactly what this animal was like and we can recreate it exactly. So there's not, the chances of, unwanted mutations or unwanted transgenic genes being thrown in there. There's also work being done that, similar to what you're saying, is taking, if we have only like I think it's the northern white rhino where there's only two individuals left. It's a mother and a daughter. And for a population like that, you want to preserve their genome, you want to preserve the mother and daughter's genome, but it's also not too late to find recently dead individuals. There were more white rhinos only a few years ago. And if you found those carcasses or preserved specimen and you could look at their genome because it wouldn't be degraded. You wouldn't have to reorder it, make a transgenic organism. It's recent enough that you could fully find the genome of that individual. And if you can do that, then you can essentially clone a dead individual and bring back that one. You could use a southern white rhino, or sorry, a black rhino as a surrogate, and then create essentially a clone of a long dead rhino, and that would bring back genetic diversity into the population. Because if you have two southern white rhinos left that are female and they're both, it's a mother and daughter, the population is essentially gone. You just have these two individuals that can't breed with anyone and are going to die out. But by using these tools that you're talking about of preserving the genomes of, you know, nearing extinction species, then you can use it to bring back biodiversity and save the population before it kind of goes over the edge. Uh, yeah, I hope that answered your question. Thank you for the question.
Thank you very much. Yes.
All right, thank you, Gene. And next up, we have Bill Tharaway. Go ahead, Bill.
Hey, Russell, can you hear me?
Yeah, we can hear you.
Let me see if I can get my face on here. Can you see me? No, I can't get. I can't get the video off.
Oh, we
There
can
we
see
go.
you now.
Hey, how you doing? Oh,
Hey there.
before I get into my question, Graham, I want to compliment you on the seamless connection you made between your talk and your graphics.
Thank you.
I try to do that all the time and never quite succeed as well as you. So
Thank,
nice job, nice
I
job.
appreciate that so much.
Okay. Back in the day, I used to work for the Army doing research in their Natick labs,
Mm,
and I was working on Kevlar, which
mm,
a protects you against bullets.
mm.
The and on the team we had biologists, and what they did, they took the gene that expresses a protein in spider's web and
Yeah, I'm familiar.
put it into a goat, and they were
Yeah.
getting goat's milk, and they were getting enough of this protein that they were able to synthesize huge amounts of spider web. Turned
Mm,
out that it wasn't that good for Kevlar, but since it was biologically um, uh, compatible, it's now used all over the world. Anytime
mm.
doctors want to sew something up or something like that. So some of the things turn out to be all right. As
Yeah.
far as the ethical questions, I'll just leave you with one thought. It
Mm,
was written on a bathroom back in the day when I was going to Columbia University, and it goes like this: if we're going to play God, we need to get good at it.
yeah.
So that's my suggestion: is
Yeah.
that like all science, you can't, you never seem to, you don't get the whole thing all at once. You
Mm,
stumble along at a little at a time. As far as genetic engineering, that train has left the station,
mm.
and we need to just try to catch up. Just
Yeah,
try to get good at it. I think that's the most
exactly.
important thing.
Yeah, I mean, a lot of science is based on principles of trial and error.
Yeah,
And when it comes to transgenics, what's key about it is it's permanent change. That change gets passed down to your offspring. So once it enters the gene pool, it's there. It's not going away.
yeah,
And if you're playing God, you also have to have the, what's the word, omnipotence. Like you have to have the full understanding of the world before playing with anything like this. I think transgenics has been used in a lot of good ways. It saves thousands of people a year, transgenics does. We don't realize it, but anyone who's diabetic, your insulin is transgenic insulin.
yes, yes,
Exactly. It's transgenic. And, so much of our crops being grown now are not just genetically modified, but transgenic. If you go, it was a big issue in Canada and a little bit in the US where transgenic organisms are currently being sold in supermarkets. There are species of fish, I think it's aquaculture fish, where it says salmon, but it's actually three species of fish mixed into one. It's salmon, it's another salmon that grows really quickly, so it'll grow fast. And it's a type of eel that can survive in cold environments. So it's the perfect animal to grow, up in the north. But you're also putting the risk out there of what if these salmon escape their environment and get into the gene pool? You've made a permanent change to the wild population before you even knew what you're doing.
yeah,
So yeah, I appreciate that point. Like before you play God, you have to know what you're doing because
yes.
trial and error isn't trial and error if that error is permanent, you know?
Yes. I see your point.
Yeah. But I very much I think that's kind of the whole crux of this argument of, you know, do we really understand what we're doing? So yeah, I thank you for your point.
All right, thank you, Bill. I see a couple more hands. If Graham, if we're okay to keep going a little bit
Oh yeah.
longer. Next up would be Sandy. Go ahead, Sandy.
Can you hear me?
Oh yeah, I can hear you.
Okay. First of all, thank you. You gave me so much to think about that it hadn't even occurred to me on
I'm
this
glad.
topic. But the other side of it is, it just seems that for every scientific, I don't know, discovery tool, whatever you want to call it, there are people who do it for bad reasons. I mean, you're talking about the consequences, the unfortunate consequences that could happen when people are doing things for good.
Yeah.
But there's also people who do things for bad reasons.
Mhm.
And can you comment on any of that?
Yeah, I think that, you know, transgenics, or you can expand on it as just genetic engineering as a whole, is a Pandora's box that we do not realize it's already been opened. I think it's important to note, aside from this talk, that genetically engineered people exist currently. That when this science was first done, there was a Chinese scientist who, in secret, and the government of China was very much aware of the science going on in the country, and still he snuck under the radar and created two genetically engineered children, or a bunch of genetically engineered twins, all of which were aborted except one pair that were born, and they were born with a gene that gave them a increased resistance to HIV and AIDS. And, that was a good thing. But at the same time, it was only after that happened that that scientist, he was arrested for doing this in the first place, but it was only afterwards that it was released that, oh, these genes that we thought controlled HIV resistance may actually influence intelligence. And these two kids, it's these two little girls who exist today. They're like 3 or 4 years old. They have, altered genes that are compiled from different individuals, and it gives them a leg up. They're smarter than everyone. Like they have an increased intelligence. And yeah, you make a really good point of we talk about so much, bad genetic engineering that when you see what I'm showing you of like good genetic engineering, you realize even the good side is really flawed. So yeah, I would say that it's a scary science, and I think what we should all keep the biggest eye on is how easy this is. It's a very easy thing to do. As I showed you at the beginning of the presentation, I took a 2-week course in molecular cloning, and in 2 weeks, I made a transgenic bacteria. I gave it the ability to glow green and glow red, but you could give it a ability to be ampicillin resistant. You could take the resistance in every different type of bacteria. One's resistant to ampicillin, one's resistant to penicillin. If you put those together and make a transgenic bacteria, it could be the next biggest disease on the planet. Um, so yeah, I think that we all need to be aware that this Pandora's box is open, and we need to examine how ready we are to have this part of our world because it's already out there. It's just kind of a matter of, is it happening privately and we're not seeing it? Is it happening in closed doors? Who's in control of it?
And you're what you're pointing out is with those bacteria and what people could do is people with bad intent. I mean, there's
Exactly.
enough problems with the good intent, but
Exactly.
people
Yeah.
also, to use this for something frivolous,
Hmm.
that's another issue. Because as you mentioned, um, I don't know which organization it was that's trying to
Hmm.
recreate the woolly mammoth. I mean,
Colossal biosciences.
why do we need a woolly mammoth?
Yeah. I mean, I showed a little
That,
bit. Oh yeah.
I mean, that's just frivolous.
Yeah. I would agree with you in a lot of that. I think that, bringing back these extinct species, the idea is that it would reestablish order in an ecosystem, because if it's a recently extinct species, that environment is still suffering from the loss of its recently extinct member, like the mammoth is, if you went back 10,000 years, a lot of Siberia was grassland because the mammoth would knock down trees there. But nowadays, without the mammoth, it's all forest. And that actually surprisingly sequesters less carbon than a grassland would. So there are benefits. There is something to be said about there's a clear reason that this could work. But just because it makes sense in our minds doesn't mean it's actually going to turn out that way. I would want to add one last thing. When I made the transgenic bacteria that were bioluminescent, we also gave them ampicillin resistance. So in 2 weeks, made a glowing, you know, resistant bacteria. So if it's so easy, we need to really consider, can anybody do this?
Well, I can't.
Take a 2 week class and you'll get it. I guess that's a scary thing. It would take you 2 weeks to understand it and do it yourself.
And I was a chemistry major, so
Yeah. So probably just one, yeah. Thank you for your question. I appreciate it.
and thank you so much for the presentation.
Of course, thank you for coming.
Thank you, Sandy. Next up, we have a question from Alice. Go ahead, Alice. And Alice, you'll have to press unmute one more time.
I lost that. Okay. I come from a regulated industry,
Hmm.
and it kind of concerns me that people aren't thinking through everything. Particularly with the people from Colossal Bioscience. They get a lot of VC capital.
Hmm.
They get a lot of people who have bells and whistles,
Hmm.
and there's no regulatory environment surrounding this. It's just a
Exactly.
free-for-all.
But there's also the question of who would be the regulatory party.
Well, yeah, and the problem is that today, at least, as long as you have money, you can get regulatory compliance, right?
Yeah, completely agree.
Which is absurd.
Mm.
And it's getting worse. When I was in the elephant exhibit, people were getting very excited about, the de-extinction of woolly mammoths. And I said, and where would you put them?
Yeah.
Not only that, elephantids generally are herd animals. They need a herd.
Yeah, and they're so social that they need to learn from one another.
Right.
Yeah.
So again, they get all the bells and whistles, they put it out on TV, but where does it get regulated? Agriculture? FDA? Which who does it?
Yeah.
Someone,
That's a great question. I guess I want to throw a question back at you of who do you think should be in control of that? Because, should it be, if I'm trying to bring back like a mammoth or something, should it be the people who, and if I want to put that mammoth in Siberia, should it be the people who live in Siberia that control it? Should it be the scientists? Should it be their government? Should it be the human race as a collective,
That's a question that no one can answer right now, at
Yeah.
least.
Yeah.
But the regulatory, there is no regulatory environment for this.
Yeah.
It's a free-for-all.
Yeah. With the coral that I mentioned, the transgenic coral that's resistant to changes in salinity and temperature.
Yeah.
I think it was in 2023, around there, or 2022. After only a few years, some Australian labs, I think around Sydney, got approval to start testing out their transgenic coral, not in a lab, but in the Great Barrier Reef.
Well,
They
I
were-
actually have experience working on drug development in Australia,
Hmm.
and it's one of the most lax,
Yeah.
frightening
Yeah,
regulatory environments I've ever worked in.
and to be so relaxed about coral, it's one thing for if the US was very relaxed about, or let's take if Mongolia was very relaxed about their policies, maybe I'm wrong. I think, man, a country that has no coral reefs around it, they can be, their rules don't really matter. They could get approval to do something because it's not that important. But Australia, if it's such a lax process, that wouldn't be a big concern if it wasn't for the fact that so many coral reefs are in Australia. They
Well, they
may,
depend upon the Great Barrier Reef.
yeah, but well, the thing is,
Critical.
they depend on it, but also the whole world does. All
Yeah.
of our fisheries across the planet depend on it. So while it's, they're making decisions about what's happening in their own, land, whatever, or what's happening in their own waterways, yeah, sure, it can be left up to them because it's their, it's their country, but it's something that affects us all. And yeah, it's like, should they be able to make a decision like that would influence the whole world? It's a good question.
I mean, Mongolia did allow the Przewalski horses to come back.
Yeah,
But
yeah, exactly. I'm not going to put Mongolia in charge of coral reefs, but I'm not going to put Australia in charge of, Mongolian horses. It's a really complicated issue of who should be in charge of
yeah,
this because everybody has an opinion.
yep,
Uh, so yeah, I very much appreciate that question. That's exactly what I was, hoping to get to with this
yeah.
presentation.
Thank you.
Thank you.
Right. And so I think that's it. I don't see any other hands. Thank you all for jumping in and sharing your thoughts, questions, and ideas about this topic. And once again, Graham, thank you so much for giving the talk in the first place.
Oh my goodness. Yeah, thank you so much. I very much appreciate giving me this opportunity. I really enjoyed it. And I really appreciate everyone who showed up.
Likewise. And so on that note, this will be our last Zoom for the calendar year, but we resume in January with a couple of talks already scheduled. So I hope all of you will stick with it and join us again as those January talks are announced. But before then, everyone, if I don't catch you, have happy holidays and a happy new year as well. And stay warm out there, everyone. All right. Have a good afternoon. Bye.
All right. Thank you all. Thank you.